Hi, folks, and welcome to Lesson 4 of the Unit 1 in the Higher Human Biology course. Today, we're going to be looking at the topic of genes and proteins in health and disease, in other words, mutations. So let's kick off with the definition of a mutation at higher level. We learned about them being a random change in the genetic material at Nat5. Higher, there's just a slight alteration to that. We say mutations are changes in the DNA that can result in no protein or an altered protein being synthesized. Okay, so either no protein or an altered protein being synthesized. And the reason for that is because when there are mutations or changes in the DNA, that will then resultingly change the codon, which means a different amino acid will be brought to the ribosome to be synthesized into a different protein. Okay, so changes in the DNA mean a different codon, a different amino acid, and therefore it will change the protein. We'll either get no protein at all, or the protein will be altered in some way. And we'll talk about how that happens throughout this lesson. So we're going to be looking at different types of mutations and how they impact the production of proteins. So we're going to kick off with the single gene mutations first of all. Now, single gene mutations are ones which involve the alteration of a DNA nucleotide sequence. A base, nucleotide base, is altered in the sequence, and that can either have minimal impact or quite a large impact depending on the type of alteration that occurs there. So we're going to look at three different ones, and we're going to look at the varying impacts of each of them as well. Now, if we look on the screen here, we can see that we've got a normal DNA strand here, okay? GAG, TGC, ACT. And when we convert that into mRNA and we read the codon at the ribosome, these produce these different amino acids, okay? We're just calling them amino acid 1, 2, and 3 for simplicity. There are names for the amino acids, but you obviously don't need to know all of them for the exam. So I'm keeping it nice and simple here. Now, the first type of mutation that is a single gene mutation will be a substitution mutation here. Now, a substitution mutation is where you replace a nucleotide base with another within the sequence, okay? So in our original sequence of DNA here, we start off GAG. In our substituted mutation, which is right here, substitution being right here, we see that the A that was in this one has been replaced by this T here. So it's been substituted. Now, if we look at the rest of the sequence, all the other letters remain the same as they are in this original DNA at the very top here. Okay, so there's only that one nucleotide base that has changed. Now, the impact of that is that it will alter one amino acid. Okay, it alters the amino acid that is coded for by this codon, but it does not impact the subsequent amino acids. They remain the same as they were. Okay, so substitution, where we're replacing one nucleotide base with another, has the impact of changing one amino acid, one codon. Okay, that's well worth remembering. The second one we're going to look at is insertion. Insertion, as the name suggests, involves adding a nucleotide base to the sequence. So we look at the original DNA here. We've got GAG, it starts. Now, if we look down at this one, this goes GT. So we've got an extra T in here. Okay, this T has been inserted into the sequence. So above here, it was replacing the A, but here it's been inserted and the A and the G are still there. Okay, they've just been moved along. Okay, all of the letters, you can see all the way along, all of these bases have been moved along one place. Okay, if we compare them to the original DNA. Now, the impact of that is that not only do we change the amino acid that's coded for here by this first triplet of bases, we then change every subsequent amino acid that comes after as well. Okay, so that now codes for a different amino acid here, and this one also codes for a different amino acid here as well. Okay, so that's insertion right there. And the last one we're going to look at is deletion. Now, deletion, again, as the name suggests, involves removing a nucleotide base from the sequence. Okay, so this one here is going to be our deletion. And again, see if you can spot what's been deleted. And we can see from the original sequence that that A is no longer there because the original sequence started G, A, G, T. And on this sequence, we've just got the G, G, T. So the A has been removed, okay? So there has been a little removal of that A from in there, okay? Now, the impact of that is that all of the nucleotide bases have moved again, but this time they have moved in that direction, okay? whereas they moved in the other direction before with the insertion. With the deletion, they're all moving one place in that direction. Now, again, what this means is not only does the codon and the amino acid change for this first triplet, the codon and therefore the amino acid for every subsequent one also changes. Now, if we compare the amino acid sequence that makes up the first protein here, 1, 2, 3, and we compare it with substitution, we can see only one of the amino acids is changed. When we look at insertion, we can see 1, 2, 3 of the amino acids are changed, i.e. all of them. And when we look at deletion, also 1, 2, 3 have been altered and produced a completely different protein here. Now, we call those insertions and deletions, we call them frame shifts, okay? We call them frame shift mutations. Now, the reason we do that is because what we are doing with these mutations is we are shifting the frame, we're shifting the sequence in some way, okay? In this one, we're going that way, and in this one, we're going that way, okay? So we've got our insertion here, shifting the frame to the right. We've got our deletion here, shifting the frame to the left. Now, an interesting story about how that name came about, the frame shift. In old movies, they used to have a film reel and it had individual frames, okay, individual little pictures on the reel. And because they spun the reel so fast, those individual frames or pictures looked like a sort of live action movie. Now, when they were editing those reels, what they used to do was literally cut out a frame, or if they wanted to add in something, they would add in a frame, okay? They would insert or delete frames within the reel. And the impact of that is, obviously, if you inserted a frame into the reel, then all of the scenes in the film shifted forward a few seconds. And similarly, if you removed a frame from the reel, all of the subsequent frames moved backwards in the reel and would have occurred at an earlier point in the film. Okay, and it's the exact same for our DNA sequence here. Okay, frame shifts cause all of the codons, and all of the amino acids after the mutation to be changed. Okay, so that's a frame shift mutation, and that involves both our insertions and our deletions that we've learned about. Okay, so we call them frame shift mutations. What is the impact of this? Well, as you can guess from the fact that it changes every codon and every amino acid afterwards, it has a major effect on the structure of the protein that's produced, okay? It'll be completely different, and it'll either be a different protein, it'll be a non-functional protein, or even it might even code for no protein at all, okay, depending on how severe that change is. So that's frame shift, which includes insertion and deletion. Now we're going to talk a little bit more about substitution mutations, because we briefly mentioned them at the start about changing one nucleotide base, and that affects one amino acid, okay? Now, nucleotide substitutions are split into three main groups. We've got the missense ones, we've got the nonsense ones, and we've got the splice site mutations. Okay, and we're going to talk about how each of those three substitutions are different from each other. So a missense mutation, that's this one here, results in just one amino acid being changed for another. So in this case, the original sequence has amino acid 1. When a missense substitution happens, that replaces amino acid 1 with amino acid 4. And if we look at the rest of the sequence, we see no change in the amino acids there. Okay, so we've just directly changed one amino acid for another. Now, this has the impact of perhaps resulting in a non-functional protein, okay, one that doesn't carry out the function it is supposed to, or sometimes it can have little effect in the protein. Just one amino acid might not change the protein significantly and it can still do its function. Or we even have this scenario where, because our codons, often we have more than one codon coding for the same amino acid, it means that even when we change the codon, sometimes it might code for the same amino acid, so it might not even cause a change here. But anyway, we'll either have a non-functional protein or have little effect on the protein with the missense ones. Now, for the nonsense ones, okay, and that's them here in the diagram, they result in a premature stop codon. It's well worth remembering that whole phrase, a premature stop codon, which is being produced. And what that does is it results in a shorter protein, okay? And what do we mean by that? Well, for example, if the G from the original strand here is replaced by a T, substituted with a T, That then codes for not another amino acid, but for a stop codon. And we know from the previous topic that a stop codon will instruct the ribosome to stop translation. So therefore, the protein will end here, okay, or the polypeptide chain will end here. And the result of that will be that we get a shortened protein. So it's the premature stop codon, which results in a shortened protein. That's what this substitute mutation does, the nonsense mutation, okay? You might think all of this just sounds like nonsense, to be honest, but these are the names that are given to these substitution mutations. Okay, and the last type of substitution mutation is called a splice site mutation. Now, those result in some introns being retained and some exons not being included within the mature transcript. So if we remember back to the previous topic, we looked at RNA splicing and we talked about removing the introns and retaining the exons. Now, if there's a mutation at a splice site, i.e. between an exon and an intron, okay, in these areas here, if there's a mutation in those areas, that will mean that the cell won't be able to work out what to remove and what to retain. And when we have no mutation, we normally get exon, joined together, intron removed. But with the mutation involved, sometimes we will retain an intron in the mature transcript, or we lose this exon here, okay? And that is not retained. And we need that exon, and we don't need that intron. So we're ending up with mature transcripts here, which are not going to code for the right protein, okay? They are not going to have the right codons to make the amino acids, which make up the protein that we actually want. So splice site mutations result in some introns being retained, or some exons not being included within our mature transcript. Okay, and on to the last wee bit of this topic where we talk about chromosome structure mutations. Now, chromosome mutations can involve changes to either the chromosome number. Okay, remember we have 46 chromosomes within the human cell normally, but there can be changes to that if there's a chromosome structure mutation. And also changes to the structure, which is what we're going to focus on in the next little section here. So there are 4 different changes to the chromosome structure that can occur. And one of them actually shares a name with our single gene mutations, which is deletion. Now, deletion, and remember this for all your definitions for chromosome, you have to say is where a section of a chromosome is removed, okay? Remember that bit, section of a chromosome. So deletion is where a section of a chromosome is removed. And if we look at the diagram over here, we can see this C gene here, okay, this C part of the chromosome. When we go over here, that has gone, it's been removed, okay, it's been deleted. So deletion, removing a section of a chromosome. The second one, duplication. This is where a section of the chromosome is added from its homologous partner, okay? Now we know from the first topic that all of our chromosomes are in homologous pairs. So in duplication, we are getting a section added from the homologous partner. So if we look at this example here again, we've got A, B, C, D, E, F, G, H. That's fine. That's all normal in that chromosome. But in the duplication error, the C and D from the homologous partner have been added to this chromosome, okay? If we looked at the homologous partner, we'd see it lacked the C and D because it's gone over to this chromosome here, okay? So it's added from its homologous partner. That's duplication, okay? And you can see visually there how that works, CD, but then CD again. And they've got that extra C and D from the homologous partner. The next one is inversion. And if you imagine, if you invert something, if you invert your hand or you invert a chair, whatever, you're turning it upside down, you're reversing it, okay? So this is where a section of a chromosome is reversed. And what we can see here is if we look at the normal sequence, A all the way through to H in the normal order, when an inversion mutation happens, we can see that the B, the C, and the D have in fact been inverted, haven't they? They have been inverted and the whole thing has swapped round. Okay, it's like it's been flipped upside down, if you can imagine it like that. And the last one is probably the trickiest one for people to get their head around, is translocation. Okay, now trans as a prefix in biology we know means to move to or to cross over to, and translocation means to move to a new location. So this is where a section of the chromosome is added to a chromosome, but this time it's not its homologous partner. Duplication is the homologous partner. Translocation is not the homologous partner. So if we look at this, A, B, C, D, E, F, blah, 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 and then we've got different letters here to represent the genes on the other chromosome, we can see that the A and the B have moved. Okay? They were on the top chromosome and they've moved down here. And the I, J, and K were down here and they have now moved up to this one. So a section of the chromosome has been added to its partner that it's not, and it's not its homologous partner. How do we know it's not homologous? Because it's got different letters, different genes, different sections on this. It's at a different length as well. Okay, so this is not its homologous partner that it has added a section of chromosome to. Okay, so keep those four in mind and try and remember the difference between all of them. The names give you a hint, but you need to know these wee definitions as well. And note that they all start with a section of a chromosome. Okay, so you have to mention a section of a chromosome when you're describing the change in a chromosome mutation. Now, what does this mean? Well, it means that the substantial, and it is substantial changes, remember, we're not just changing a single nucleotide base, we are changing large sections of a chromosome. Okay, so we are changing a lot. Now, the substantial changes in the chromosome mutations often make them lethal. Okay, so they are more lethal than the single gene mutations that we've already been learning about. Okay, and again, just keep that in mind as you might be asked about that in the exam. So that's the end of the lesson part of this this particular video, and we're going to now move on to looking at some past paper questions that are involved with this topic. Okay, so let's have a look at some of the past paper questions on our topic that we've just covered. So we'll start with some multi-choice just to get us warmed up. So as usual, you can pause the video when I've read the question if you want to have a go at these yourself, and then you can hear the answer and explanation afterwards. So number one, the diagram below shows two chromosomes M and N before and after a chromosomal mutation. The form of mutation that has taken place is a... Okay, so let's look at what we have got here. Okay, it can either be a translocation, a duplication, an insertion, or deletion. So look at what we have. We've got M and N. Okay, that's two chromosomes. Immediately, what can we see? Well, we can see that M and N are not homologous. Okay, they are not homologous. So therefore, we can immediately eliminate duplication. And then when we look at what's actually happened to them, gene X and gene Y have been swapped, okay? Gene Y has been added to N and gene X has been added to, okay? Which means that this is going to be an example of translocation because these are not homologous to each other. These are different chromosomes, okay? They're not homologous pairs. So translocation for that one. Number 4. The following are descriptions of 3 single gene mutations. Okay, we've got the exon, intron, codons are repeated or destroyed, one amino acid is replaced with another, and one amino acid codon is replaced with a stop codon. So which line in the table matches the descriptions with the correct gene mutation? So let's have a look here. As soon as we see exon, intron, we should be thinking splice site. Okay, splice site mutations. As soon as we see one amino acid codon replaced with another, we should be thinking missense. And once we see amino acid codon is replaced with a stop codon, we should be thinking this whole thing is a load of nonsense. So which line in the table below matches the descriptions with the correct gene mutation? Right, so number 1 we've worked out is splice site. So splice site being number 1 is option D. Description 2 is missense, okay, 2 missense, and 3 is nonsense. So the only one that works from our options there is D. You need to know these descriptions for each of the substitution mutations, so make sure you learn those so that you can identify them as quickly as we were able to as we did that exam question there. Next one. A genetic disorder of the nervous system results from a mutation in which a nucleotide is inserted into a gene. Which of the following types of mutation causes this genetic disorder? Okay, so this is a insertion. Okay, it's been inserted into a gene. So what's that going to be? Well, nonsense we know is a substitution mutation. Missense, also a substitution mutation. Translocation is a chromosome mutation. That's where a section of the chromosome has been altered. This one is where a nucleotide is inserted into a gene. Okay, we know that insertion is a type of frame shift mutation, so our answer is going to be D there. Okay, number 4. Which line in the table below shows the effects of the different types of gene mutations on the protein formed? So we know that a nonsense mutation, that has a premature stop codon, which results in a shortened protein. So that means we can have that one and we can have that one. So it's going to be A or B. In this sense, we know replaces one amino acid for another. Okay, so proteins contain one different amino acid. All the amino acids change from this point. That's not right. And we know that these are both nonsense as well, so they're both wrong too. So we can eliminate C and D there as well. And then frame shift. Well, we know with the frame shift that it affects every amino acid after the mutation. Okay, every amino acid after that point. Okay, so that's going to be A there, because all three of those are correct. The next one, which type of gene mutation occurs when a codon for an amino acid is replaced by a stop codon? As soon as we think stop codon, we think shortened protein, and we think this is all a load of nonsense. Okay, next one, individuals with Cre2-Chat syndrome have a shortened chromosome 5. No other chromosomes are affected. Which type of mutation causes the Cre2-Chat syndrome? So shortened chromosome 5, it means it has had part of its chromosome deleted if it's shortened. And that part of the chromosome has not gone to any other chromosome because it says no other chromosomes are affected. So this has to be deletion here. Okay. If it said that another chromosome had become longer, then we might be thinking translocation, or if it said it was homologous partner, we'd be thinking duplication. But because no other chromosomes are affected, that means it's got to be deletion there. Next one, which of the following terms describes types of mutations which occur in both genes and chromosomes? Now, we know that one type occurs in both the single gene and chromosome, and that's the deletion one there, okay? That is chromosome only, that is chromosome only, that is single gene only, that one. Okay, this occurs in both. Which the following list is a single gene mutations. We've got nonsense, missense, and frame shift. Which of these gene mutations is the result of a single nucleotide substitution? Okay, so frame shift, that involves insertion and that involves deletion. Okay, it does not involve substitution. So we can rule that one out. Missense, that is where we replace one amino acid for another because of one nucleotide base being replaced with another. So that is substitution. And nonsense, we know that's where we've got a premature stop codon, which is due to changing one nucleotide base and replacing it with another. So also substitution mutation. Okay, so the answer is going to be 1 and 2 there, which is going to be B from our options. Okay. And then we've got this extended response question where I have to write notes under those headings. I'm going to do extended response questions in a separate video rather than these videos just because they take quite a long time to do. I'm going to leave that one for just now, but I hope going through those questions was useful for you and that you can leave me a comment if you think that there was something else you'd like me to cover or anything that you thought was particularly useful there. Okay, folks, that's all for this lesson. Class dismissed.